Hello everyone, it's Dr. Demi here, and in this video I am going to be speaking about paper 3. I'm doing this because of popular demand, first of all, and also because I realized that quite a number of students are homeschooled and may not have access to laboratory facilities, and I would like to bridge that gap to the best of my ability so that you are somewhat prepared for the exam. The best way to prepare for paper 3 obviously is to do practicals, but even if you can't do them, understanding some of the theory might help you when you go into the exam. So I'll give you some tips in this video and also do a sample question, which is just questions 1A and B of the June 2019 version 3 paper, so that you have an idea of what to expect and how you can approach some of the theoretical problems that you will encounter. So I hope that you find it useful. Let's get into it. So first things first is I want to point out the skills and knowledge you need for paper 3 as well as some extra things that you need to know. So first of all, you need to understand what serial dilutions are and how to calculate them. Understanding serial dilutions will go a very long way in paper 3 because I realized that over the past 3 years this has become sort of a constant feature in the questions the students get. So knowing how to do serial dilutions is important, and I'll just go into it in a bit of detail in this video, but I intend to do another video on how to do food tests, serial dilutions, and so on in more detail. You also need to be able to do food tests like I've just mentioned. Food tests are things like being able to test for starch, being able to test for proteins, how to test for reducing and non-reducing sugars, as well as how to test for lipids. So that are the 5 classes of tests that you are expected to know. And usually you're not given the instructions on how to do them. You're just expected to know how to do them. So I will do a video on how to do that. It's not going to be the most animated or perfect video, but I hope it gives you an idea of how to make it work. Then you need to be good at data presentation. Data presentation basically refers to how you present whatever data you get during the experiment, either in tables or in graphs. This gives you marks simply because in paper 3, they do not draw the tables for you, and most of the time they don't draw the graphs for you. You are required to draw the graphs, label the axes, and when you have a table, have table headings that are very clear as well. So being able to do that is something that you need to know for paper 3. You also need to know enzymes and the factors that affect them. That's very important. And I'll explain that in a bit, but the next thing you should know is extrapolations. So when you draw a graph, say for example you have a graph that looks like that from your experiment, and then they say find the value of x based on the graph that you've drawn. So you should be able to come in and say, okay, if x is sitting somewhere here, then on the y-axis it correlates to this value, and then extrapolate. So that's basically what an extrapolation is. You also need to be able to do biological drawings. If you don't know what biological drawings are, please check out chapter 7, the playlist that I have on chapter 7. I believe if I did a short video, really short video on how to do biological drawings there. Please check that out because this is important. And now, if you haven't been listening to anything I've been saying, please listen to this part. Whenever you do paper 3, you will come in contact with 2 questions. Paper 3 is 40 marks in total for 2 questions, and those 2 questions are for 2 hours. The way it works is that each question, so if you have question 1, A, B, C, D, that whole of question 1 is only for 1 hour. So it's 1 hour per question. Once 1 hour has passed, you will be required to move to the second question, which would be a different set of experiments or a different topic. So always plan your time well whenever you go into your paper 3. And I'll explain this a bit more with food tests because I think that's where students tend to lose a lot of time. Always plan your time well when you get into the exam, because you know you have only 1 hour per question. Now, in terms of the combinations of questions that you get, you will always have a question on biological drawings. That is a constant that we have seen in paper 3, and there is nothing that indicates that that is going to change anytime soon. So biological drawings will be one question, and the second question will either be on food tests or biological molecules, or on enzymes, which is chapter 3. Okay, so this is chapter 3 and this is chapter 2. So in terms of the topics that you should learn for your exam, I would say learn enzymes and the factors that affect them, how to do food tests, and any other understanding about the biological molecules, as well as biological drawings. Of course, you might get extra questions that require you to maybe answer things that relate to transporting plants or transporting animals, but the core of the experiments are usually based on these three topics. So if you can do these three topics, then you should be fine. If you couple that with your skills on serial dilutions, data presentation, and extrapolations, then you are more likely to get full marks in paper 3, which I'm sure is what most of you want. So as an example now, let us look at this question here. It says milk protein concentrates, MPCs, are used for manufacturing foods such as cheese and ice cream. And then it just gives you some more information that there are different concentrations of proteins in different types of milk. And they then go on to say the concentration of protein in milk can be measured using potassium hydroxide solution and copper sulfate solution. When I do the video on food test, I will explain a little bit more about this, but this is often called the Biuret test. Okay, so you might get into the exam and they don't give you copper sulfate and potassium hydroxide, but rather they give you what is called a Biuret solution. It is the same thing. You add the Biuret solution to the protein mix or to the unknown solution, and you'd be able to tell if it's a protein or not. And I think I have a slide about that that shows that in a bit. Then it says you need to prepare a serial dilution of the proteins in the milk M, and you need to carry out the test for protein on each concentration of milk. So this is what you're given. When you get into the lab, you will find that if you were doing an experiment like this, there is a rack on the table, like a wooden rack or a plastic rack, and in that rack you're given test tubes. Okay, you're given test tubes, and the test tubes will be marked as such: M, W, P, and C. And obviously you will have extra test tubes, and I'll explain what those are for later. But as they've told you, M is 100% milk, and they've given you the volume of M. They've also told you that W is distilled water. They've given you the volume of distilled water, and you have 5% potassium hydroxide solution for that much, and you also have copper sulfate solution for that much. Then they tell you that if any solution comes in contact with your skin, make sure you wash it off immediately under cold water, and you also wear suitable eye protection. So this is not stuff that you necessarily have to worry about, as your school should. Hopefully they do this. They ought to give you gloves when you get into the lab, and also give you a lab coat as well as eye goggles. So if they don't give you those three things, please ensure to ask for them. Gloves, eye goggles, as well as a laboratory coat, so that you don't hurt yourself when you are busy. So let's look at what they want us to do. They want us to make this cereal dilution of 100% milk M. And they say the concentration of the milk, so the concentration needs to decrease by a factor of 10 between each successive dilution. And then figure 1.1 shows the first two beakers you will use to make your cereal dilution. So remember that you will have beakers, you will have test tubes, and all of these things before you. For those who don't know, a beaker looks something like that. Okay. I think I'll just show you images of this in the videos that I do on food tests and so on. Beaker looks something like that, and the test tube is the longer one. Obviously, I'm terrible at drawing, but I hope that this gives you an idea. So they then say complete figure 1.1. So this is the first thing you have to do for marks. Complete figure 1.1 by drawing as many extra beakers as you need for your cereal dilution. Now, something I'm going to tell you is that the general rule, if you're doing a cereal dilution, is to draw 5 beakers, or to make 5 beakers of dilution. All right, most of the time students do not exceed 5. So as long as you do 5, then you should be okay. Then they say here, under each beaker, state the volume and concentration of M available for use in the investigation, and I'll explain what that means in a bit. Use one arrow to label above the beaker to show the volume and concentration of M added to the concentration, to prepare the concentration, and another arrow above the beaker to show the volume of water added, because water is W. So let's look at this figure so that you understand better. So this is the image that we've got, and over here they're telling us that they put 10 centimeter cubes of 100% M. So if you remember at the beginning, we were given 25 cubic centimeters of 100% M. So of that 25 cubic centimeters, they've basically said, this is probably not been provided to you. So what they're saying is that take 10 centimeter cubes of that, or 10 cubic centimeters, and add it into beaker number A. We're going to call this beaker number A. Okay, add 0 centimeter cubes of water, all right, into beaker number A, and then over here they're telling you that there's 9 centimeter cubes left to use. What this already tells you is that it gives you an idea of what you're expected to do next. So first things first that I would do, remember I said you draw 5 beakers. Okay, so you draw 5 beakers and make sure you follow the same pattern that they have shown you. Do not try to draw arrows that look like this if the arrows they've shown you are going that way. So make sure you follow the convention that they've already started. And this is for 3 marks. So you get marks, for example, for making sure that your drawing is accurate. But these are the other things that you get marks for. Now look at this here. It says 10 centimeter cubes of 100% M and 0 centimeter cubes or 0 cubic centimeters of water have been added. And then they say 9 cubic centimeters of 100% M is left to use. What that tells you is that they have taken 1 cubic centimeter from this over here, 1 cubic centimeters from that, you can see the arrow there, to add to the next one. And if the final volume is going to be 10 cubic centimeters, if the final volume is going to be 10 cubic centimeters, that means you add over here 9 cubic centimeters of water. Okay. Now, obviously, I've done this way to show you that CIE actually already tells you in a way what you should do. Now, in this case, something else that you want to state is that if I'm adding only 1 cubic centimeters out of 10 into 9 cubic centimeters of water, and make sure you write water or W, whatever the label is, just so that you are clear on what it is that you're adding. So 1 cubic centimeters of M, 9 cubic centimeters of W. What you have done is that you have reduced the concentration of M that you're adding here by a factor of 10. And remember what they said is that the concentration reduces by a factor of 10. A good way to think about that is to say, well, if I'm starting with 100% and I'm reducing by a factor of 10, it means that you divide it by 10, and so the next concentration is actually 10%. I hope that makes sense. So it means over here you have 10%, 10 cubic centimeters of 10%. Just going to write it here, 10 cubic centimeters of 10% M. All right, and obviously from here you repeat the same step. You take 1 cubic centimeter into the next beaker, which I haven't done properly. I'm just going to redraw that so that you're not confused. You take another 1 cubic centimeters, you add 9 cubic centimeters of W, okay, and that means you're now dividing that 10% by 10, and that gives you 1%, and so you write on top here 1% of 10 centimeter or 10 centimeter or 10 cubic centimeters. Sorry, I learned how to say it as 10 centimeter cube. 10 cubic centimeters of 1% M. Okay, I feel like I'm just writing in a hurried manner here. So for this third beaker, it's 10 cubic centimeters of 1% M. Okay, I hope that makes sense. This is basically how you do a serial dilution. Another way that people do a serial dilution, if you're trying to find the dilution factor, I'm just going to say this quickly, you can use dilution factor is equal to the initial volume over the final volume. So if you know both the initial volume and the final volume, you can calculate the dilution factor. In case you don't know that, you can also in some cases use C1V1 is equal to C2V2. All right, and you can sort of use this to calculate how much volume should you be adding into a second solution in order to make in order to make the concentration right. So in this case, for example, we know that we started with 100%, and here we have V1, and we know that the dilution decreases by a factor of 10. So for our next solution, we know that the dilution is going to be, or the concentration rather, is going to be 10%. And we know that the total volume has to be 10 cubic centimeters, right? So in this case now, we can say that 10 times 10 is 100. So that means it's going to be 100. I'm just going to erase this beaker over here so that you can see. So it means it's going to be 100 V1 is equal to 100. I hope this makes sense. And in that case, V1 then becomes 100 over 100, and that is 1 cubic centimeters. So in this case, you've now been able to calculate that from the first beaker, I need to take 1 cubic centimeters in order to make a final volume of 10 centimeter cubes and to reduce my concentration to 10%. And obviously, when you take a final volume from there, you're left with 9 centimeter cubes left to use, 9 centimeter cube of, or 9 cubic centimeter of 10% to use, 10% M to use. Okay, and you do the same thing, but what you need to bear in mind here is that when you get to the last beaker, you're not going to take anything out of it. So you won't have 9 cubic centimeters left to use. You will have 10 cubic centimeters in that last beaker. So if you're doing 5 beakers with this example here, you can already calculate from the beginning. I'm just going to erase a bit here, that if I'm reducing the dilution factor by 10 each time, you can do this on like scrap paper. Please don't do it as part of your drawing. You say 100 divided by 10 gives me 10. 10 divided by 10 will give me 1. 1 divided by 10 gives me 0.1. And 0.1 divided by 10 gives me 0.01. So this is 1, 2, 3, 4, 5. And those are the 5 concentrations that you will then need to do for your serial dilution. I hope this makes sense. I know I spent a bit of time on it, but I hope that it's clear. I'll try again to explain when I do the video about food tests, how to do food tests and how to do serial dilutions, but I hope that this makes sense. I'm not sure if I ever did a video on how to do serial dilutions, actually, now that I think about it, but perhaps I did, who knows. So here they then tell you carry out the test for proteins on different concentrations of milk using step 1 to step 9. So remember, you've made 5 different concentrations of milk, and now they're telling you to test the concentration of proteins in each of those ones. So they say prepare the concentrations as decided or as shown in figure, and as shown in figure 1.1. So basically based on the serial dilution. Use a glass rod to mix the milk solution and the water together. So you'll be given a glass rod in the exam. Most of the time, actually all of the time, your class or your teacher, your school will provide you with all the things that you need. So don't worry about taking a rod of glass from home to mix your solutions together. You don't need to do that. So they then say label the test tubes with a concentration. So this is something you should have done already when you were preparing the dilutions, just so you know which dilution is which. Okay, so you should make sure that you have done the concentrations separately. Also, something else I just want to point out, when you're doing a serial dilution, when you're going from one solution to the next one, make sure you either use a different beaker or you wash the beaker. Not the beaker, you use a different syringe or you wash the syringe, but I'll explain this again later when I explain the tricks of doing a serial dilution. Then they say label the test tubes, put 1 centimeter cube of each concentration or 1 cubic centimeter into the appropriately labeled test tube. Label another test tube 0 and put 1 cubic centimeter of water, and then you add these into each of the test tubes. So whenever you get into paper 3 and you get instructions that are this long, I always tell students read all of these instructions first before you do anything. So don't just read instruction 1 and then rush to do that and then do instruction 2 and then no, read everything to the end, because that helps you work a lot faster actually. It might seem like you're starting off slow, but you end up picking up really quickly. So in this case, we've already done this because that was what we were required to do in the first stage when we did the serial dilution. Okay, so they said prepare the serial dilution according to what you've drawn. and obviously mix them with a glass rod. Then you have to label the test tubes obviously as you're preparing. So it means you label the test tubes as 100%, 10%. So that means you go to the lab with a marker. So we also have a test tube that's 1%, we have 0.1%, and then we have 0.01%. Okay. And they say in each of these test tubes, so you take test tubes and you label them. So in each of these test tubes, you take 1 cubic centimeter from the beakers. So remember you did your serial dilution in a beaker. Now you're going to take 1 cubic centimeter from that beaker into a test tube. So that means you will have 5 different test tubes, all of which will be labeled with these concentrations. Then you have a 6th test tube where you have 0.0%. And in that 6th test tube, all you have to put in is water. Now in each test tube, you add 1 cubic centimeter of P, which is your potassium hydroxide, and a cubic centimeter of C, which is your copper sulfate solution. That's basically what they're saying, and you shake it gently. You leave them to stand for 2 minutes. In this case, you can use a stopwatch to measure what you're doing in terms of time. You leave them to stand for 2 minutes, and then you shake the solutions again, and you record your results. And then they've told you that a blue color indicates that the concentration of protein is too low to be detected. Now I put this here just to show you what it would look like. That when you do a protein test, a negative test is basically one that is blue, because what that tells you is that there is too low a concentration of protein, or there is no protein at all. What this means is that in your test tube that you labeled W, or the one that was 0.0%, where you put just water, the solution should come out blue. If in the process of your experiment the solution comes out purple, it means that you had contamination in your syringe, which is why I explained that whenever you are doing these kinds of experiments with serial dilutions, make sure you either use different syringes or you wash the syringe each time, so that you don't get concentration contamination, and you don't get contamination in general. So I'll explain what I mean by that. Let's say, for example, we have prepared our beakers over here. I'm just going to do 3 beakers. So let's say we have 10%, we have 1%, and we have 0.1%. And now from these beakers, I want to take solutions and put them in test tubes so that I can do the protein test. So I take the solution from beaker number 1, which is the 10% one. I use a syringe, so I'm just going to draw the syringe as a block here. I take 1 cubic centimeter from here and I put it in here. That syringe, if I don't wash it and I come into the 1% solution and I take it and I put it in the 1% test tube, what that's going to do is that whatever residue is left from the 10% test tube will now mix with the 1% test tube and affect the concentration. So it means the concentration I'll be putting in will not actually be 1%. So whenever you're doing this, if you're provided with not enough syringes, make sure you have distilled water, and you can ask your lab for this. You take an empty beaker with just distilled water and you use that beaker to rinse your syringe. So you clean out the syringe. So it means you take from the 10 centimeter or 10% test tube, you put in the 10% beaker rather, you put in the test tube, you take the syringe, you wash it or you rinse it with water, and then you go into the 1 centimeter one and you transfer into that test tube, you rinse it again, and then you do another transfer. Please do this so that you don't run the risk of, first of all, contaminating and having to repeat your experiment. Because if in this case you go from here without rinsing and then you get to the one that's 0%, you also haven't rinsed it, and you pick up the solution of water and put it in a 0% test tube, there's a chance that when you test the test tube for proteins, you might see that it turns purple, which then indicates that you've had some level of contamination. So that's just something I'm trying to point out to you here. Okay, then they say here, record your results in an appropriate table for the known concentrations of milk. So obviously in this case, we don't have the experiment running, but I want to show you how to present the table. If you look at this question, they have something about the key, and I think I'll just paste the key here so that you can see. It's not on the video as I'm recording now, but I'll paste it after recording. But here, in this case, they say record in an appropriate table. So you have to be the one to draw the table, and you get marks for, first of all, having a good heading, like good headings for the different parts of the table, and also recording different concentrations. So in this case, because we are simply testing the concentrations of milk, I would say concentrations of milk. I'm not going to write it in full, or just write concentrations of milk. And then in brackets, put the unit. The unit is percentage. Please always put the unit on the heading. If you don't do that, you will lose marks, okay? And then in this case, you can put perhaps color change, because what you're observing with the protein test is actually a color change, and you have to record that using the key. Now, again, we've done 5 different dilutions, right? Or we've started with 5 different solutions. So we can say 100, 10, 1, 0.1, 0.01. Okay, so we have those. And what would the color change be? Obviously, if you have 100% of milk, then you will have a lot of positive marks. I'm basing this on the key. I can't remember what the key says, but whatever is the maximum change on the key would be for 100%. Okay, but you can't just say that. You have to observe it as well. So that means it needs to be really purple. So you can use the key here, and you might also want to add color observed, if that, but that depends on you. You don't necessarily have to do that. And then you can say maybe deep purple or something, deep purple here, or something like that, okay? And then you do the same for 10. But if they've said specifically that you have to use the key, then make sure that you use the key. Also, something you will get marks for here is showing that the trend in the color change is consistent. So for example, you can't have for 100 a color change that is not that deep. Maybe it's just something like 2 positive signs. But then at 0.01%, you have a color change that shows like a really, really deep purple compared to that one. And then at 0%, you also have a positive sign. That already is an indication that your trend is off, and you will lose marks for that. So make sure that your trend is correct when you do this recording. Okay, so for this part of the question, they are asking you an extrapolation question. They say that after doing this experiment and interpreting with the key and all of that, you're provided with a different type of milk X, and the concentration of protein in X is unknown. You now need to carry out the test for protein on X and use your results to estimate the concentration of protein in X. So this is the extrapolation thing I was telling you about. So first things first, they tell you what to do yet again. They say label a test tube with the letter X, put 1 cubic centimeter of X into the test tube labeled X, and then repeat steps 5 to step 7, which is basically adding your potassium hydroxide solution as well as your copper sulfate solution, and then you leave it to stand for 2 minutes. So you basically repeat the same thing that you've done for the first 5 experiments that you had. So for the 100%, for the 10%, for the 1%, for the 0.1%, and so on. So you're repeating the exact same thing. And they say after step 12, record your results for X using one of the symbols in the key shown in figure 1.2. So say, for example, you record, you look at the figure and you see that, well, I think I'll just estimate X as that, okay? They then say the 100% M contains 12 grams of protein in 100 cubic centimeters. So 100% of M is 12 grams of protein, okay, in 100 cubic centimeters. They say using your results in this and this, estimate the concentration of protein X. So in this case, what you might want to do is look at what you've gotten as your result for X and look at which value amongst all of these correspond to this. So remember, when you did all of these concentrations, you also got, you know, the key. You also used the key to interpret. So let's assume 10% was also like that. That means what that's telling you is that there is 10% of protein in X, right? That there's a correspondence between those two. So what that it says is that if 12 grams is 100%, 10% you can easily estimate to be 1.2 grams. Okay, and then you can sort of say X is then 1.2 grams of protein per 100 centimeters. Well, I hope that's correct because I've just realized I'm really exhausted and my brain is falling asleep. But yeah, that should explain how you do this question. So it's usually very easy, and it's usually a lot easier when you're given a graph to do it. And on the next couple of slides, I think I have a graph there that sort of shows how to do a bar graph but doesn't really explain the extrapolation factor. But being able to extrapolate, very important. Being able to present your data in the table, also very important. Something else I think I should just add here very quickly before we move on is that whenever you do your table and you have your headings for your table, so heading 1, heading 2, you get marks for the headings, by the way, for using the correct headings, and you put in the units for each heading. I've also seen that sometimes they like it when you draw the lines to differentiate the different readings for the headings. So, for example, if this is solution 1, and your heading here is the concentration, 10%, and the heading here is color change, and you say color change is that, they like it when you use lines to delineate that very clearly so that they can see what links to what. And I believe this is because students tend to write roughly. In drawing the tables, you use a pencil to draw the table itself, but you use a pen to fill in the table. So that's something else to bear in mind. Okay, this is another question that you will always get when you do paper threes. They ask you about the errors. What is one significant error that you need to know? So in this case, think of the way the experiment has been conducted. You have to rely on your ability to distinguish color, okay? Because you're mixing all of these solutions, and the point is you want to see a purple color, and you want to differentiate between different shades of purple to determine the concentration. So one source of error obviously would then be identifying the color change. So you can say color change identification over here. My pen. Okay, so you can say color change. I'm just going to write changes that identification. And the explanation there can be that there might be difficulties or errors in judging the different shades of purple. Then they ask you, this is also another question, and if you can get these kinds of questions right, then most of the time you can get really good marks. They say suggest 3 improvements to this experiment so that a more accurate estimate of the protein concentration of X can be determined. So if you want to make things more accurate, think of how you've run this experiment from the beginning. You started off with trying to figure out the concentrations based on the key for a range of values, and those were like decreasing by factors of 10%. But what if in order to determine X, you only had to decrease your dilution factor by 5? So you can talk of making more dilutions, okay? So making an increased number of dilutions. So I'm just going to write it as making more dilutions. That might help you. Dilutions with less or with smaller dilution factors. Smaller dilution factors. That's one advantage, one improvement that you can make. Another improvement is related to the error that you've already identified here, that you have to identify the colors and be able to tell the differences in the shades of color, even though you're not necessarily someone who deals in colors, you're just a biology student. So in that case, you can suggest the use of a colorimeter. A colorimeter is an instrument that is used to, like the name suggests, measure color, and that would give you a more accurate distinction in terms of what the colors are. Okay, another thing then is that you might want to test X at the same time as the others. So test X at the same time as the other dilutions. And the reason you might want to do that is that you want to keep all the conditions of the experiment the same. And you also perhaps want to allow for easier comparability of the samples. Because think of the fact that you do your tests on the 5 different dilutions that you made, and then while they're still standing there, now you have to do X. What if the reaction in them still continues, and so the shades change while you're busy with X? So it makes sense for you to do X around the same time so that it is easy for you to then observe what is happening. Okay, now this is the last slide, and this is basically again data presentation, but just wanted to point out to you that whenever you're asked to draw a graph, they will give you this type of graph sheet. Um, what you need to do, they say, for example, a student carried out some research into the percentage mass of protein in milk from different mammals, and they show you the different mammals as well as the percentage mass of the protein. Then they say plot a bar chart. So in this case, they've been very specific. For those who might not be aware, this is a bar chart. Ugh, that's such a terrible drawing. I will try to do one that's better. Okay. So a bar chart is the one that looks like this. Okay. That is a bar chart. For those, a line graph is something else where you basically plot points and then you join them together with a line. So those are two different types of graphs that you can come across, but there are obviously many more that you should familiarize yourself with. So in this case, what you will get marks for is labeling the axis first of all. Do not assume that because CIE has provided you with a table that they should sort of intuitively know what you're trying to represent. Make sure you label your axis very carefully. For example, in this case, because the numerical figures or the numerical values here are only one thing, then we have to put those on the y-axis. That's an easy way for you to remember it if you're dealing with something like this. And on the x-axis, you then put the different types of animals, such as the buffalo, the camel, and things like that. Then you have to be very clear on how you are counting for your data. So if this is point 0 over here, then you need to count the squares, for example, to determine which one should take me to number 1, which one should take me to number 4. So typically, I would often say that students should just count them as maybe 0.1, especially if you're dealing with decimal values. So you can say 0.1, 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.6, 0.7, 0.8, 0.9, and then 1. And then just mark that as 1. Because what that will help is for you to plot a value like 0.8, you can easily just count then this is 0.1, 0.1, 0.2, 0.3, 0.4, 0.5, 0.6, 0.7, 0.8, and you can say, okay, this is my 0.8 over here. So I'm just going to draw my bar for the buffalo. Obviously, in terms of the width that you can take, that depends on how many values you have here. Like you can see over here, we have the buffalo, the camel, the horse, the goat, and the sheep. So there are 6 of them. And we don't have to write them out in full because they have put in brackets some labels there that we can use. So for convenience sake, you use the label. So you can just write them as B, C, H, and so on. And you draw your labels there. So for things you need to do, label your y-axis. Your y-axis would be percentage mass of protein, okay? And then your x-axis is type of mammal. Make sure you put that there. The other thing you need to do is to put the values for the axis, especially the y-axis where your values are supposed to be. So you do that again by doing the counting, and you can, when you do your counting, this is 1.0, and you do the same counting. Now I'm just going to estimate 2.0, and then 3.0, and 4.0, like that, okay? So by doing it that way, then you're able to justify why your bars are sitting the way they're sitting, and make sure you use a sharp pencil to represent your bars. Also, the last thing I would say here before I end this video at a 40-minute mark is that when you draw your bars, make sure that they are only as wide as what your letter is covering. Don't write your letter C here and draw the bar for C like that, because that will cause confusion and cause you to lose marks. So yeah, that's it from me for this video. I hope you've enjoyed it. Have fun. Goodbye.